Ow. Guess it's time to go back to work.
Good evening. Good evening. Thank you. Welcome to the Shoals Marine Laboratory's weekly marine science seminar. I am Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Lab, and I want to thank you for joining us this evening for our sixth Rock Talk of the 2024 season. Our seminars are affectionately called Rock Talks, in part because back in the early days of the lab, the seminars and classroom lectures would occur in the intertitle On the Rocks. Although now we are meeting in the Commons, these weekly seminars provide a wonderful opportunity for our island community to come together to learn about a range of topics related to marine sciences and to our environment. For those of you who are joining us online who may be new to our lab community, the Shoals Marine Lab is the oldest and largest marine lab in the country that is focused on undergraduate training, both in coursework and in research. The lab is operated jointly by Cornell University and the University of New Hampshire. In addition to our guests that are joining us online tonight, we have 3 courses on Appledore: Marine Environmental Science. Y'all are being quiet tonight. Marine Ecosystem Research and Management, and Shark Biology and Conservation.
Oh, nice.
We also have our summer undergraduate research group, affectionately known as SURGES, who are involved in a summer-long research project. We're excited to have everyone both in person and online joining us tonight for our seminar. The format for tonight's seminar will be a 45-minute talk followed by 15 minutes of questions and answers. For our friends online, please use the Q&A box at the bottom of your Zoom screen. We will read your online questions out loud for the entire audience. For those of you in the Commons, we'll pass around a microphone for questions from you so that everyone can hear. This evening's speaker is Dr. Christopher Siddon, who is the Chief Scientist for Marine Fisheries with the Alaska Department of Fish and Game. Chris completed his BA in Biochemistry and Molecular Biology at Hamilton College. After graduating, he found the light and spent the next year in the East-West Marine Biology program with Northeastern University. This program involved an intensive and experiential marine science education through classes and independent projects in Friday Harbor, Washington, Discovery Bay, Jamaica, and Nahant, Massachusetts. He then completed his PhD at Brown University studying trait and density-mediated indirect interactions in the New England Rocky subtidal zone. He has served on the North Pacific Fisheries Management Council, the North Pacific Research Board Science Panel, including 8 years as chair, the Pollock Conservation Cooperative Research Center Advisory Board, Alaska Sea Grant Advisory Panel, and the Alaska Ocean Observing Systems Board. Chris's Shoals connections include not only completing the bulk of his graduate research here between 1997 and 2003, but he has also served as the mentor to the Rocky Intertidal Transect interns and served on the faculty of Field Marine Biology and Ecology and currently on Marine Ecosystem Research and Management. He also met his wife on the island. It is my pleasure to introduce my colleague and friend, Dr. Chris Siddon, who will be presenting his talk, The Aleutian Island Golden King Crab Fishery, a case study in large-scale cooperative research. Please join me in welcoming Dr. Chris Siddon.
Thank you very much, Sarah. That was a great introduction. Thanks everyone for joining us here and online. Hopefully my audio is fine for you online. I get a thumbs up from Matt, so that's great. My first question, as Sarah read my title, who has a comment on my title? I do, it's way too long. Take that into advisement. Yeah, it's true. Excellent, right? So again, we're always learning. I'm just like, I did that. I'm like, that needs to be improved. So that's fine. Thank you for that. We will get into it. I decided today to start with our conclusions, our take-home points. Let's start right off the bat and get to what we need to talk about. Point number 1 that I'll try to make tonight: we don't always get it right. We never get it right on the first go. Pilot data, preliminary data are essential in what we do in marine science and in science in general. 2, quote unquote, non-significant results can really be super important, right? We have to think, oh, that was not significant. We start doing stats, we start getting into experimental design. Oh, that's not significant. P-value is not less than 0.05. That's terrible. That is not the case, right? Depends on our question, and having a good question matters. And I'll show you an example of that tonight. 3, you all are receiving all the foundations right here for doing large scale marine ecology. And I focus here on including the social, community, and the communication aspects. We can all learn the foundations of what a crab is and what an experimental design is and what a sample size is and how to do stats. But the interactions of people and the communication skills, whether it's oral or writing, are absolutely essential. And you're gonna do that here as well as learning the content. And those things are as important, if not more important than the content. And this is a great place to do that. And last, there's really lots of really cool marine ecology left to do. We get into class and we're like, oh, someone already did that. Someone already did that. I can tell you there's lots of really cool stuff to do. And if you don't think so, come talk to me and we'll come up with some really cool things. I got lots of projects in mind. Okay. Quick disclaimer and a quick warning. I'm going to show a video clip later in the talk. It's only a couple of minutes, but I'm going to show you a 30-second clip now. If anyone is super motion sick, watch this 30-second clip and see how we want to deal with that. Let's find my cursor. We're gonna want 30 seconds. This is the boat that I was on. This is some pilot data that we got for some of the talk I'm gonna give. You do get motion sickness, not a big deal. We're gonna do the opposite. Usually they tell you to watch the horizon. I'm on the boat taking the video. Don't watch the horizon because the horizon is what's moving a whole heck of a lot, right? So this is a vessel out in the Aleutian. So I'll show you a little bit more on this in a little bit. Just be forewarned, you're going to see a couple of minutes of boat rocking, but we'll see some crab. And we do it for a reason, not just for watching the crab boat rocking. Okay, good. Not so bad. Who's seen The Deadliest Catch? Okay, right. We've seen video like this. Okay. So a quick outline of what I'm going to talk about tonight. The first part is just the background, and we're going to go through this relatively quickly. I know there's a bunch of bullets here, but we'll do this relatively quickly. I'm going to talk about population genetics. I'm going to talk about fisheries data. I'm going to talk about survey design. I'm going to talk about king crab, and then I'm going to talk about the golden king crab fishery very quickly, just as a backdrop. And hopefully we'll tie all of those pieces together for the most part of the center part of the talk. And that's really to talk about doing cooperative research with the commercial fishing industry, right? And I'm going to give you 2 case studies here. One is a golden king crab stock structure where we got funding to do some population genetics of golden king crab. And the second is a fishery facilitated survey. So how to do actual stock assessment survey utilizing and working with the commercial fishermen. Generally, we think about the commercial fishermen and the managers and the scientists being a little bit at odds with one another. Here, this is where we really need to work together to do what we need to do good science for everybody, the scientists, the managers, and the industry at the same time. And last, we'll do the take-home points again in case all y'all forgot. Population genetics. Since I'm from Alaska now, we should talk about salmon just for a minute because it's the easiest one to think about. There are 5 species of salmon, Pacific salmon, right? Chum, Chinook, coho, sockeye, and pink salmon. There are 32 principal river systems in Alaska for salmon. There are 264 separate stocks with escapement goals that we follow. All of those are separated, can be identified and separated by population genetics, by genetics. So we know them. So within a given river, we can have the same species, but different stocks because they have different timings and different genetics. So this is population genetics, knowing one stock from another. at the population level is essential for how we do fisheries management. Without it, we would very likely over harvest a given stock if we didn't know it was its own genetic unit as opposed to a whole species, right? So that's super important. So this is why in almost all fisheries, we really work to have the genetic structure, the population structure to know what those are. So we know if it's a stock, one stock versus another, if it's 264, if it's only two, if it's only one, right? So then we can focus our management at the stock level and not the population level, at the species level, right? Okay, that's it for PopGen for a background. Fishery dependent data. Fishery dependent data. What, anyone have an idea of what fishery dependent data is? Yeah. Depends on the fishermen, right? They're the ones that are actually, they're not collecting the data, but the data is coming from what they are doing, from their activities. Total catch per unit effort. You're going to hear that a lot probably tonight and over the next couple of weeks. CPUE, we'll use it. I won't usually think about it, but we'll say CPUE, catch per unit effort, catch per pot, catch per trawl, catch per hook. That's all just a general term, CPUE. Number of vessels, right? You have 10 vessels out doing the fishing. Do you have 100 vessels out doing the fishing, right? We need to know those are dependent on the fishery. We also have at-sea observers in a lot of fisheries. We have folks that are paid to go out on commercial vessels to measure lengths and ages, sex ratios. This is also fishery dependent data. So this is a picture, a map in the Aleutians, and all the green dots here are where pots were placed over the last 20 years. They set a lot of gear, right? It's pretty amazing. Fishery dependent data is amazing because they do it for a living and they're on the water all the time, right? Pretty impressive amount of information. Here's a similar map, similar place in the world. All the open boxes are places that were not fished, have not been fished since 2004. And the boxes colored in green are ones that have been fished since 2004 and onward to 2015. So currently only 30% of the total area is being fished. Historically, 75% of the catch came from those core green boxes, right? And historically, the CPUE, the catch rates, is 60% greater in the core area, right? So this is an interesting issue because the industry goal is to catch as many fish or crab in this instance as fast as they can, right? That's their goal, no bones about it. What happens is this leads to a CPUE, catch per unit effort, that is biased statistically. They are trying to keep their catch rate as high as possible. So if it starts to go down, what do you think they do? Fish somewhere else, move, right? Totally. If fishing was great in this spot, I'm going to put my gear back in that spot. When it starts getting not so great, I'm going to move it somewhere else, right? That leads to a bias in the CPUE data that we get from fishery dependent information. And therefore, it's a less informative as an index of abundance. Generally, we think of as CPUE catch rate goes up, we assume that the abundance of the population of the stock goes up. As that goes down, we assume the abundance is, it's an index, right? But if that index is now biased because they're trying to keep their CPUE at the same level, then it's not doing what we expect it to do. And that becomes a problem. And one of the major problems. The other thing about fishery dependent data, there's lots of gear. We just talked about that. So their sample sizes are super high. There are lots of vessels so they can spread out and they cover all of the really good areas very well. And the last one is super important as well. They're super knowledgeable about what they do. They don't necessarily speak the same with the same terms, but they know the water, they know the animals, they know their boats and how to do things. And to not tap into that information is a big miss. So it's super important to do and being able to communicate that. On the flip side of fishery dependent data are fishery independent data. And this is what we generally think about in survey design when we go out to design a survey, how to quantify how many animals are in the ocean, right? And we generally, for most commercial fisheries, we try to do this. The potential bias of fishery dependent data that we just talked about is too big of a concern from our standpoint, trying to manage a stock for long term. It's too big of a potential bias to overlook. So we really worked hard to move to fishery independent data. Here's a picture. This is the Eastern Bering Sea of Alaska. Each one of those boxes is a 20 by 20 nautical mile grid. Pretty big. Let's see. The whole area is about 500 kilometers from the coast of Alaska offshore and about 1,000 kilometers from north. So it's a pretty big area. And we just split it into 20 by 20 nautical mile boxes. The depth from shore to the depth to deep is from 50 meters to about 200 meters, right? So, I mean, deep, but not super deep in the grand scheme of fisheries. And what National Marine Fisheries Service does here in this survey, they do a trawl. Mike, you can remind me, 30 minutes, 60 minutes these days? 30 minute trawl, dragging net on the bottom, catch everything that they can in the middle of each one of those boxes. They systematically, and I have that systematic sampling, they systematically go through every box and do a 30-minute tow. The tows are big, but they're not a mile or a kilometer wide. I mean, each one of those boxes is 20 kilometers by 20 kilometers. So the footprint of that is pretty small, but it's systematic. We have a mathematical way to understand that, and we do it repeatedly over years. So now we can have we're more likely to be relying on that index to track the abundance of the animals. Most commercial fisheries rely on fishery independent survey design. But there's always a smaller sample size than in fishery dependent data. We generally try to focus on multiple species because it's super costly to run vessels. And so we want to not just focus on cod, we want to focus on 5 different flatfish species, another one, and we can try to do that all at the same time. What do you think that does? Slows things down. Any other ideas? All fish don't live in the same exact types of habitats, right? So now we're doing a broad brush. It's a Swiss Army knife as opposed to a scalpel that really wants to go in and look just for pollock or just for Pacific cod or just for, right? Because they have different requirements, right? So that's a little bit of a problem. And the fishermen generally don't. They're fishing for a single stock, a single species, maybe a couple, right? But so they're really honing in on what those species like. And there are a lot of good skippers out there that do surveys, but I'm going to say they do not have the wealth of knowledge as the skippers that do it year in and year out for those species. Okay, that's it for fishery dependent and independent data and survey design. And this will all come together in a couple of minutes. We're going to move to king crab. This is a red king crab, and that is me a handful of years ago in one of our surveys. Decapods. What's decapod mean? 10, thank goodness. There you go. Right? So prawns, shrimp, lobsters, mud shrimp, hermit crabs, and crabs. Any idea where king crab fit in? Fruit crab, that's a guess. Anyone else? It's a good guess, but it's not quite right. They're more related to hermit crabs than they are the true crab. So the hermit crabs, the Anamurin, porcelain crabs, squat lobsters, sand crabs, the Lithodidae, which are the family of king crabs, and then hermit crabs, and then terrestrial hermits. There's your quick evolution of the decapod systematic. There's a lot of super cool stuff to do in crustacean systematic. So projects, think about those if you're interested. For king crab in the Lithodid family, there are over 130 species. They're generally deep and cold water species. They're found in the North Pacific, the North Atlantic, New Zealand, Southern Africa. They're all over. This is also, I want someone to do the evolutionary tree and understand how related these things are, and the over evolutionary time, how do you get to all these different places when you think about warm water at the equator? And these are not warm water species. But I think that would be a super cool, interesting project. Many of the king crab are commercially important. Red crab, blue crab, and golden king crab for Alaska. Red, blue, and goldens in Alaska, all circled. There are 3 in Southeast Alaska, red, blue, and goldens. There are a number of red king crab stocks in Alaska, circled by red, right? This should just check in, and 3 different stocks of blue king crab in Alaska, and then 3 stocks of golden king crab. And we are going to focus today on the Aleutian Island golden king crab. Okay, so that is one separate fishery for us. Just a transition, so we're going to go into the golden king crab fishery. And this, we are going to move now and just watch a little piece of them, of the fishing operation, so you guys get a feel for what it's like today, a couple of minutes in the life of some golden king crab fishing. So this was a trip that I took a handful of years ago as one of the first trips out. I got permission to go out on an Aleutian Island golden king crab boat. I had done some commercial work with red king crabs, so we needed to get out and see if we could work with the commercial crew. And here comes the first pot up and over the side. The pots are about 500 to 600 pounds a piece. There's usually about 4. There are 5 guys on deck right now to handle the whole thing. They long line these pots. So there's about 35 pots, and they're all about 200 meters apart on the bottom. So the whole string of pots is like 2.5 miles long. They'll bring the gear on deck, open it up. You'll see the tray in the middle come across, and they'll empty the crab out into the tray. And it's not really crappy out, but it's pretty crappy out, right? You can see the boat rolling pretty good in all directions. They're dumping the crab out. They're using a big broom, right? Okay, so this is the same one, but it's just at a different angle so we can get a little bit closer. Those same pot's gonna come up and over the side. Again, careful watching the horizon when it's there. There you go. So this is probably 8 to 10 foot seas. Do you notice anything about the guys on deck? They're what? I can't hear you. They're not strapped down. That's true. Right? Anything else you notice? They got colors on. That is also a good thing. What's that guy doing? Standing on the rail of the boat, right? They may or may not have life vests on underneath their jackets. They probably do. A lot of them use CO2 inflatable lights. When you hit the water, they pop up, right? Dump all the crab, and then they'll sort all the crab. This is a very typical pot. And you'll watch them, so he's pushing them over. All this time, the next pot's coming to this surface, right? So they only have a couple of minutes to get through this. They're measuring. They got a stick, measure if it's legal or not legal. The ones that are legal are going towards the upper right into the hold. All the other ones, they're getting down a chute, a nice ramp, a slide back into the ocean. They're amazingly careful with the animals, right? It's 8 to 10 foot feet, and they're trying to be careful with what they're doing, right? To me, it's pretty amazing how seemingly nonchalant these guys are and how smooth they're working as a group. And I guarantee if we all went out on this boat at the same time, we would not be as smooth, sure-footed as these guys, right? And then they plop the pot and what? 500 to 600 pounds, 8 foot seas. Here we go. and run it to the back. And for the birders, there's some other bird. If you have noticed that you've been eyeing them in the background, good, excellent, awesome. And then the next pot comes, and that's it. And they get to do that 12, 16, 18 hours a day. It's pretty amazing. But that's, so this is from a science standpoint. I'm gonna move to the, why did I show the video? The professionalism of the crew, right? It's like you're not just going out on a boat that are just yahoos out there, you know, like the deadliest catch, they're all like screaming and being crazy, right? They're super professional, all of, you know, don't get me wrong, they like they're a little bit of adrenaline junkies, but they're super professional. You can see it's like, it comes with having a positive interaction with the skipper and the crew. The only way for me to be out there is to have a good interaction and to be able to work right next to them. You know, I was only a couple of feet. I didn't show it here, but I'm taking crab off those and measuring and sexing crab and doing that. And what this was also a reminder for me to tell you that this was a first trip. This was pilot data. This was so essential to do the couple of projects that I'm going to show you next, because it provides real information for me to see how the deck works, how I can do a science experiment in amongst 4 or 5 guys running around trying to make money in high seas. How do I do science? How do I do an experimental design on that platform? Okay, so now let's move to some cooperative research with all of that backdrop. What's the biggest problem in the Aleutian Islands? And it's geography. There's no question about it. The Aleutians are 1,400 kilometers in length across the whole chain. Here shown, they're about 75 kilometers north to south. They range in depth from 50 meters to 1,000 meters is where the Aleutian Island golden king crab. Remember the survey I showed you for the Eastern Bering Sea goes from shore to 200 meters. These guys are fishing down to 1,000 meters, and it goes to that depth in 75 kilometers, as opposed to 500 kilometers. This is a mountain range versus the plains. This is a whole different environment, right? And so that requires a different way to do things. Rocky substrate, there's strong tidal currents here. When I started in my current position about a decade ago, there were no genetic data for this stock, for this, none. We've been having a fishery on it since the 50s, so 70 years, 60 years at that point in time. We didn't know if this was one stock or 264 like salmon, but we've been fishing it consistently. And we're like, no, we need to start doing something about that, right? And we were only, for the stock assessment, we were only using fishery dependent data. Just talked about how dicey that is. I mean, it's useful in some regard. And this was a decade ago, right? So this is stuff that we're like, we need to do better. The biggest thing is because of the space and the geography. It was cost prohibitive how we all would think about designing an experiment or designing a survey. We couldn't do it. There was no feasible way for me to charter a boat at $15,000 a day that run and set here over 1,400 kilometers and do all that. So we work with the fleet. Pilot data, back to pilot data and fleet interaction. Because of that trip and a couple other trips, we can show that we can collect data in crappy weather. It's a huge important thing to know if you can do or not. We can not interfere with deck operations, also super important if they're going to let you on your boat to do research. And then word gets around. People are people, right? It doesn't take long. Something happens on the island. How long do you think it takes for the island to find out? Not very long. Heed that on those that you didn't know that. It does not take very long. And all of a sudden, with those few things, a huge, large-scale project is now possible that was not possible for decades. So we did a population genetic analysis. This was a colleague of mine, Stu Grant. the geneticist, and me, the person that is the liaison between doing field work and working with skippers and knowing the experimental design. We got funding to do population genetics, which was sorely needed for this with the Aleutian Island with the industry. And we wrote a grant, and the industry wrote a letter of support that said they would do this, they would allow us, they would take us, they would feed us, they would do all the things on there. So we split the Aleutians into 9 or 10 different sub areas, 8 sub areas, excuse me. We collected 917 total crab. We collected about 115 from each one of those sub areas. And then we only allowed ourselves to sample, and we were just drawing blood for DNA. Is how you do that for crab. 1 crab per pot and 5 crab per string. Any idea why we wanted to do that? And last, males and females of various sizes. So those last 2 things, why do you think we were doing that? Any idea? Yeah. Right. Good. So spread things out. Want to make sure we're sampling as most representatively as we can across the whole chain. So we don't want to. We collected 115 all from 1 pot. Like, well, are they all brothers and sisters from 1 parent? We don't want to do that if we're really trying to get an idea of the population genetics. We want as mixed, right? As many different ages or sizes, because we can't really age crab. So let's choose different sizes in a pot. Let's not just do all the ones that are this big because they all came from the same year, basically, or 1 or 2 years. Let's mix it up, right? So we're trying to spread things out to be as representative as possible. Good. And here's the short answer to a lot of genetics and a lot of details. On the x-axis is geographic distance, so farther apart. And then on the y-axis is genetic difference, distance. And the short answer is there is no statistical relationship between how far apart you are and what your genetic distance is, right? So there's no stock structure, genetic stock structure. The animals in the far east of the Aleutians and the ones in the far west of the Aleutians, 1,400 kilometers away, are not differentiated by genetics. It's all one stock. That's it for population genetics. That would not have been possible, though. Think about it. That would not have been possible if we couldn't get on board and do that with the help and the interest of the commercial crab fishery. So super amazing to be able to do that. Scientifically boring, maybe. But now we can ask why. It seems crazy that over 1,400 kilometers, there is no genetic differentiation at that level. From a fisheries management point of view, this is super exciting, right? It's like you haven't been screwing it up for the last 50 years. This is a good thing. You're right. We're managing at the correct scale, right? And thankfully, they weren't like salmon. That's a good thing. But now we know we can check that box. We've done a good job. We've done what we want to do as fishery managers and understand the stock structure of the organisms, the resources that we're extracting from the ocean. Thanks to the commercial fleet. Okay, switching gears to the facilitated, fishery facilitated survey. We're doing great. I will call it the co-op survey for short, just because that's what we've called it and it'll come out in my brain. So just bear with me when I do it that way. So really the goals, again, for the cooperative survey were to reduce the potential for what we call hyperstability, and that's having that CPUE of the fishery dependent data stay as stable as possible because they're trying to maximize their catch, right? 2 is to increase their spatial extent. Remember that, and I'll probably show it again, the green boxes, they've all kind of reduced into that core area. Like, well, if you're fishing the good spots, you might not know what's going on in those other areas. And we really want a full picture of whole population. We want to initiate a consistent time series. And 4, we want to provide a cost-effective method to do this. We priced this out to do a survey across the whole Aleutians. It was going to cost us about $4+ million, and it would take about, we did it the way we think about normally doing it. It would have taken 1 or 2 boats if we did it with a boat, it would have taken a boat 14 months to do. Is there a problem with that? We're already late, right? Like, we're trying to do the assessment for this year and know how many crab are in the ocean this year, and it takes 14 months to do it. Then we need to know they've already changed, right? Before from start to finish. So that's a problem. So here's our study design. Is EME still, they're coming on, which is kind of an experimental class, or just left. Excellent. So there's experimental design. There's classes here that do experimental design type things. So here we'll talk about experimental design. The blue in the background is the 1,000-meter contour. So we constrain our sandbox to only 1,000 meters in depth, right? And then all the green, again, are all the observer data that we have from 1990, 2015. We then basically draw 2 nautical mile boxes around every place that there was a hot set and commercial crab, any size crab caught, to try to define the habitat. In that there are 1,100 possible places that we could pick. We then, because we're scientists and we understand stratification and sample design, we think about random sampling, and you guys will hear about random sampling a lot over your tenure as scientists. Random sampling is always kind of the pinnacle of what we want to do. We want to randomly sample the environment. We don't be careful. This is a little gray. Someone might get mad at me. We don't really, random sampling gets us, it's a way for us to get to representative sampling. Sample representatively across the whole space. Random sampling gets us there when the sample sizes are high enough. Random sampling also tends to provide, you have a potential for clusters. You'll get a random sample with 3 or 4 right next to one another. And that's not necessarily representative. So there's been a lot of math and a lot of study on that. So what we do is we stratify things, and here in those 3 different color blocks, we basically say we're going to split them in thirds. We're going to do randomly select 25 in the first color, 25 in the second color, and 25 randomly selected in the third color. That makes sure we don't get all 75 that we randomly select in one area, because it could happen by chance, right? We're forcing things to spread out. So we do that, stratify by 3 areas, and we select 75. We randomly select 75 of those 1,100 areas for the commercial fleet to set their gear in. So now we've got fishers doing a scientifically sound stratified random sampling design in the system. There is no way we could do this without their ability and their willingness to change their fishing practice. What do you think what's the cost to the fishermen to do that? Yeah. Yep, we heard a lot of that. We got a lot of pushback. Like, my CPU is going to go down. What are you going to do about it? Right? Anything else? Yeah. Oh. All right. Anyone? Yeah. Travel time is a huge deal. What's travel time equate to? Money and fuel and time, right? That means they're out on the ocean longer, out, and it's like, a full day or 2-day run just to get to your gear. And so if we're making them take more time between sets of gear, which we are, it's costing them. Okay, so that's the problem. This is just to show that we've done this. We started this in 2015. They're going to get started to do this in 2024 here in the next couple of weeks. You can see on the right, all the 3 different big colors and where all the sampling, randomly stratified random sampling places are for each of those. and the number of string samples and the number of pots. This is still a super small percentage of what the fishing industry samples during their fishing. Fishing. So we still have a small sample size problem, but we have removed the potential bias of fishery dependent CPU data as an index on the weather. And this is just a modeled smooth CPUE index. So here, one could, in our class, we talked a little bit about this today, looking at a trend over time, one could argue that over this time, there's been a slight decline in CPUE over time with a slight uptick in this last year. We would never have seen that. If that's true, we would never have seen that in the fishery dependent CPUE data. Never. I can almost guarantee that. Before I talk about temperature, the other thing that was super cool about this project was the fishermen were a little cranky that we're making them fish in places outside their core areas, making them travel a little bit longer. But remember earlier I said they hadn't fished in most of those places since 2005. They went and they set gear in these random samples. They're like, this is gonna stink. And then they set their gear back there the next time because it didn't stink so bad. Right? So we are forcing them to prospect a little bit, to look in other places that they haven't looked in a long time to see. So all of a sudden now the fishermen are learning even more from us. So now we have a little bit of a two-way street of how this works. Like, oh, they know a lot, and don't get me wrong, they know a ton. Now all of a sudden, oh, the scientists, they know, they might have some, there might be something to what they're, and so that was a huge benefit. I wasn't expecting it by any stretch, but it was certainly a nice benefit from all of this. This is just a last detail to think about things. This is a temperature plot. I know it's busy. The EAG is just the eastern part of the Aleutian Islands, and the WAG is just the western half of the Aleutian. You can just see the black lines going up and down. Those are temperatures, daily temperatures, actually hourly temperatures across multiple days. And the short story here is that in the eastern half of the Aleutian Islands, the temperature fluctuates almost 2 degrees Celsius, every change of the tide. Amazing temperature differential. When you move west, it changes on the right half of that panel. It's basically less than 1 degree C every time. The oceanography is dramatically different across that whole chain. Also, is like, why the heck isn't there any stock structure, right? What else is going on? The environment, even though it's all the Aleutian chain, not all the same. The oceanography is quite different. If there's any budding oceanography here, this is a super cool question of what's going on. There are other commercially fished fish species along the Aleutian Islands as well, and so this is one of those things. Understanding the oceanography could have multiple impacts on multiple fish resource species if we have understanding of the why. Right? There's the pattern. We talked about this in our class. Mike talked about this in our class. We have a pattern, but now we need to understand, right? So then we can predict what's going on into the future. Okay, we're done. Take home points. Hopefully I hit most of these tonight. We don't get it right from the start. Pilot data are essential. Me getting out on a commercial boat to see how the operation works changes your whole perspective of what you can even think about doing as an experimental design. Non-significant, in quotes, results can be super important, right? So when you get those, think about that. And the more important thing is make sure that the question you're asking is interesting, regardless of the answer you get at the end. 3, again, you are receiving all the foundations right here. I think that's super important. Everything that I do, I've gained a good portion of them from doing work and research and interacting with the folks that are here. So that's really important. Last, my pitch. There's lots of really cool stuff to do, whether it's here whether it's in Alaska, whether it's in the world. So thank you for your time and attention, and I will take any questions that are around.
Questions?
So the first king crab study you did, Aleutian Islands into those 8 different sections.
Yep.
You didn't have an understanding of the population dynamics yet. How did you go about dividing them?
That's a great question. Did everyone hear the question? Hopefully, like, why'd we do this, the 8 sub areas? And so we did that for 2 different reasons. One, we looked at all of the effort, all of those green dots, all of the pots over 20 some years of where fishermen were fishing. And we also looked at the bathymetry and oceanography. If you look back on some of those, there are some really deep channels that cut through. And so if there were going to be any oceanographic type boundaries to the baby crab, they were more apt to be at those places that had bathymetric distinct differences. So a bunch of canyons. So we did our best guess, and then we just wanted to be able to make sure that we could split things up east to west to make sure if there was any segregation across the chain, we could tell where it came from. But that's a super good question. And that was probably one of the harder parts, is making a decision. What are the criteria for making those decisions on that? And so a lot of it was effort from the commercial fleet and the symmetry of the system. So great.
Budding oceanographer.
Awesome.
I understand that the fishermen ended up getting a fine CPUE from shifting their sample. Was there any program in place for compensation for what they were doing beforehand in case that hadn't happened?
Yeah, thanks. Compensation for them. So we also, in doing cooperative surveys in general, and do this, like trying to build in incentives to make sure that you can convince someone to do this once, maybe twice, but for us as stock assessment folks and managers, we really need a data set that's long. We talk about this in our class. Having long-term data sets are super important. So incentives are important. So the couple of things we did, just because it's fishing, you lose gear at some point, right? Something breaks, you lose gear. So we replaced 50 to 100 pots for the fleet every year to take that cost out of that. What's that slowly do? Any idea? Yeah. Standardizes the gear, right? Because they have 7 foot by 7 foot pots, and some guys use 6 foot by 6 foot pots, and some use 6 by 5.5 foot. It's like it's a little bit of a mishmash. If we tie their gear, we do it all the same, then over time it actually even gets better, right? So that was one. We've also tried to help and to pay for some fuel costs and various other things to try to do that. And the last one that hasn't quite worked that we're still working on is because it's out in the ocean, right? And it's like, and they start in August and they don't generally finish the fishery until somewhere near Christmas. That we tried to back up the start. If they were going to do the survey for us, then they could start their season 2 weeks early to help with the just better in the summer. So we're still working on that incentive because it was definitely a useful thing. But changing season dates is one of those, it's a paperwork kind of nightmare, right? It's just the way it is. So, but trying to work in incentives is super. So it's a great question.
Sorry, I just have one quick question. So I guess with your interaction with the like skippers and the different commercial vessels, I get there's the whole like burden of like them having to do the science, or execute the science. Did you ever encounter any like pushback on them just agreeing to do the science itself? Because it seems like, it's a good thing that you didn't find any stock structure because you didn't have to change anything, but it's kind of like a risk for them where if you do find something in your study, they might have to change, their operation. So it's almost like a balance between finding something out or them just kind of taking an ignorance is bliss approach.
Yep. That's super hard, right? What's that require? Oh, I heard it. Did anyone else hear it? Anyone? Trust, right? This is a dicey thing in science. It's a dicey thing in the real world, right? And it requires active participation with the people that their livelihoods matter, right? That's a super important thing. How to do that, how to be good stewards and good things. You know, it's one of the weird things. It's like these skippers that are, I would call a number of them friends at this point in time, which I would not have considered a decade ago, right? I mean, that seems like an odd thing. So yeah, that's great.
How is the fishery currently managed? Like, is it a slot fishery or is there quotas? Like how? Because I noticed that they were measuring and then the majority of the stuff they were just throwing back. Like what, how is that managed by?
Yeah, so we managed the quota. It's co-managed by National Marine Fisheries and the state of Alaska. So we basically set a catch limit for the whole thing, and then a number of participants have a proportion of that limit that they can do. And if we, the scientists, do it correctly, we set a limit that is biologically safe for replenishing the population over time. But that's where it comes, right? This stock had been using fishery dependent data, still is using fishery dependent data for coming up with that number of how many animals is the appropriate number of animals to pull out of the ocean. So we're a little, and so we try to build in some precaution to that and have some buffers along with that. And we saw, again, a lot of them putting them in the chute and sending them back. There's a size limit as well. So they have to be male and they have to be over 6 inches, 5.5 inches for this stuff. Depends on where you're at. So yeah, so there are other safeguards built into the whole thing.
I have a random question. Sorry. 1,000 meters? Is there any issue with pressure and the crabs? I'm sorry, that just blew me away.
Nope, no problems at all. They don't have a swim bladder. They come up to the surface. They do just fine. And they do it's also amazing. You saw what they do on deck. They put the legal size crab in the hold. They keep them there. They're out on the water for a couple of weeks at a time. They run fresh seawater in the hold. They bring them back. The number of crab that are dead when they get back to the dock to offload is like less than 0.5%. It's super, super small. The animals are really quite hardy. They do quite fine whether they're coming from 50 meters or coming from 1,000. Yep.
So like, I'm confused because like, if the understanding in general is that like having different stocks means different like regulation levels, like how did this specific fishery go for 70 years with the assumption that it was just one stock? And then with that question, like,
Yeah.
I assume NOAA, have they continued surveying like this to fix that assumption for other like fisheries? Like how is that allowed?
Yep. We have this long list of we really want to do's, and there are lots of fisheries that started back in the day. And once you get things started, can you imagine just saying, you can't have your livelihood anymore because we don't have the population to square it away, right? People will come unglued, right? I mean, and if things are going fairly well, like if things start to really have a problem, and those things will get more attention more quickly, right? If things are plugging along and we're like, yeah, we don't have the population genetics of pollution islands, and it's we need like, yeah, but I got 15 other fires over here. This stock is going down, and this is right. And then this one starts to, and then all of a sudden, like, oh, we need to get back to this, right? So it's really a triage to some extent, and it's looking at all the data. Yes, we really want stock information, the population genetics, but we also do know a little bit more. Salmon go to the same stream to reproduce every year. King crab have larvae and they produce, lots of larvae. And these guys actually are feeding larvae and they sink to the bottom. So there's they're a little less likely to have population structure, but way more likely than a red crab, which have larvae and they send them up into the water column and they go off in the current. So that's usually a fairly mixed, so we'd have less of a chance, but we were concerned enough. It's like, we really need to get this done. But yeah, that's a great question. There's lots of those questions. You start digging and you're like, oh, we should have data on this. And we've had a fishery on this for how long, right? There's a lot of those. You would be surprised. There's a lot of work to be done.
So are there like any worries of like inbreeding, I guess, in that sense of like them being one giant stock at that point?
No. So we would be more concerned about breeding if there was a lot of genetic structure. So I think basically it says there's a lot of mixing, even though the animals don't, one of them adults is not going to walk 1,400 kilometers, right? But so there's enough genetic mixing that's likely not going to be. Great.
Any other questions in the room?
I'm around for the next week and a half off and on. So.
Yeah.
How many days do they typically spend at sea? So like 2 weeks, and how many miles? I,
Yeah.
just curious.
So from Dutch Harbor, which is the main port, to the core fishing area that I showed most of the data from, it's about a 24-hour run just to get there, plus or minus 12 hours if you're at the eastern end or the western end, right? And so they're out for, it takes them about 12 to 15 days to run through all the gear that they have. So they have 30 pots on a string, right? We saw them haul one pot, right? Then the next one comes, it takes them a couple of hours to get through a whole string. And they do about 4 strings a day, and it takes them about 2 weeks to get through. That's how much gear they have just on one trip. And then as they do that, though, once they haul the whole string, so they've got 30 some pots on the deck, they run them all to the back of the boat. They then put them back, rebait them, reset them, and start again. And then they take their full hold and go 24 hours back to and offload all the crab, hang out in town for a little bit. But frankly, by the time they get there, they can pretty much turn around because the first pots that they brought back up and reset, it's been about 12 days because they had to get through all their gear and then get back to town. And so it's just this big conveyor belt, right? So it's on about 12 days. There's a handful of boats that fish further west, and it's closer to 48 or 36 hours just to get there. So they're out about 20 days to pop.
Just to make sure people know that around here, lobstermen are going every other day. No, I'm just blown away by this fishery entirely. It's like insane. Other questions?
So then, since they were like out for so long, were they just making like an extra trip to your like designated spots, or were they just like on their path? Or
Yeah, good. So what we tried to do is the rules, guidelines that we gave them is do this as their first trip of the season. Because that meant that the fishing grounds basically had 6 to 8 months to resettle and reorganize after they'd been fishing on them for 6 months, right? And so we asked them to go out, and they only had to fish. It was probably about half of their gear that they had to set where we wanted to, and then they had their other half of the gear that they could still set wherever they wanted to, right? So we did that on the first gig. So they basically, it took them a little bit longer, and they probably set maybe a quarter less number of strings of gear to do that. So it did cost them a little bit of money and time to do that. So, but they were actively keeping crab that were legal still at that point in time. So it wasn't such a detriment.
I want to thank Chris for a really stimulating conversation about the industry, the combination between the science and the fisheries and sustainable fishing and management. For those of you that have been around for a couple of weeks, we're seeing the 7 classes that have already come and gone are kind of combined in some of this information. And for anybody who is interested, obviously, he will still be around for the next week and a half. And you can probably find him at most meals. And so thanks for everyone who attended, both online and in person. And thanks to Dr. Siddon for being our rock talker.
Pleasure.